Greetings all, again. It's great to have you join us today. This is a hot topic. Cannabis and what we do with it and how we govern it is now on the front pages of everything. And on behalf of the panelists, I'd like to welcome you to this afternoon's session. My name is Peter Phillips. I'm the director of the Centre for the Study of Science and Innovation Policy, which is an institute jointly hosted at the University of Regina and the University of Saskatchewan in the Johnson Shoyama School, which you both in Regina and Saskatoon are now visiting. And today I'm your moderator. So I'm not the content guy. I'm the guy that's trying to make sure that everybody gets a chance to test their ideas. The speakers get a chance to say what they think that is important for you to hear, and we get an opportunity for some conversation flowing from that. We also, because we're a school with two campuses, we have the opportunity and the privilege of having our colleagues in Regina join us. So today we have at the University of Regina campus a number of people that there's probably more people than we can see because the screen sometimes doesn't always capture everybody, and that group is being facilitated by Karen Jasper Laforge from the school in Regina. So today we have a group of people who are part of CRIS, which is an interdisciplinary research team that aims to obtain scientific evidence about the application of cannabis and its derivatives to human and animal use for health, disease, and disorders. And this is a big problem, or a big subject area. Just to, this isn't my primary area, but just to give you a sense of the scale of the challenge that we're attempting to address. Keep in mind that the legislation is not passed yet. We're legalizing and regulating the use of cannabis in a whole variety of ways, and it has knock-on effects with a whole variety of actors. We don't know how many users there are. We have an estimate of about 2.3 million people in Canada, which is about half the estimate done at similar times and places in the United States, so I'm not sure we're that far apart. So it's about 6.3% of the population that are deemed to be users, mostly for recreational purposes at this point. We have 10 provinces and 3 territories, every one of which is trying a different way of putting together the governing system that will deal with the legalization and regulation of the use. As part of that, we have an estimated 65,000 police officers who are part of probably 500 or 600 different jurisdictions that each have to figure out how to adapt their practices to the emerging policy area. But what probably really brings most people to the conversation now is people think there's a large economic opportunity here. The current estimate is that the industry generates approximately $5 billion of gross sales in this country. That compares to $9 billion for beer, $7 billion for wine. So it's already a large area, but it also has a bit of a potential bubble building in the capital markets right now. There is a thing called the Canadian Marijuana Index. It lists 24 publicly listed companies. The market cap, as of this noon today, was $26 billion. The largest, Aurora and Canopy, which are each over $5 billion apiece. So this is a large play. Many people think it's a lot larger than Canada alone because of an estimated 3 to 5% of the global population are current users, and there are a lot more potential users once this gets legalized. So the CRIS initiative and people who are now attaching to it through the policy and science community and plant science community are all trying to figure out bits and pieces of this, of putting this together. Because we don't have a governing system, we don't have a science base, we don't have a commercial base that will effectively and efficiently deliver products and services. So today we have 4 speakers, right? Yep, 4 speakers who are going to speak, and we have a number of people who are collaborating with them, and I recognize people in the room who have more expertise than I do and a lot of interest in this topic. So this will be an interesting conversation. It'll be in 2 parts. First part, I'll introduce them in a moment. Each of the speakers will come up and give you a bit of a sense of where we think the problems are and what we're doing about them. Then we're going to break for a few minutes. We've got coffee perking out there that you may have seen as you come in. We'll come back, and when you come back, there will be a table up front, and we'll have a moderated conversation where you get a chance to challenge the speakers, or they get a chance to challenge each other. And at the end, we're not here to assign conclusions and decisions, but we're here to unpack some of the challenges and opportunities. So our speakers, Dr. Jane Alcorn, a professor in the College of Pharmacy and Nutrition at the University of Saskatchewan, is on the CRIS team executive project lead and is the co-leader with Dr. Robert Lepairie, who is an assistant professor in the GlaxoSmithKline chair in drug discovery and development here at the university. Drs. Huntsman and Musso from the College of Medicine are also co-leaders on the biomedical side, and I don't recognize everyone on my list, but I believe they may be in the room today and may identify as themselves yet. Dr. Tim Sharbel, a professor in the Faculty of Plant Science in the College of Agriculture and Bioresources at the university, is here and will talk about some of the plant side. And his colleague, Dr. Karen Tanino from the same faculty is part of this initiative. And then Jerome Konecsni, who some of you may recognize, was our deputy minister and head of the NRC for a while, but in his current capacity as an executive in residence in the Johnson Shoyama School, attached to the institute I run. He's going to talk a bit about the policy and governance aspects of this. So I don't think I'm going to try and explain what each of you is going to talk about. So on that, let me invite Jane up to start the conversation.
So thank you for everyone that's shown up here today. And I certainly extend a very warm welcome to our colleagues out in the University of Regina. This is certainly what we were hoping to see as a CRIS. And as you can see, this is our second CRIS Cafe. Our first CRIS Cafe was given in December 1st of last year. And it was to introduce to the campus community the CRIS. And at that time, we were a rather smaller organization, a research cluster. And from that first cafe arose the new CRIS, and as I now introduced you to that today. But that is our hope, that we continue to have these cafes so that we can have discussions around cannabis and begin to enfold all people who are interested in cannabis under a common envelope called CRIS. And so CRIS to us is a research cluster hoping to gather everyone who's interested in Saskatchewan to contribute to CRIS in the way that they think they can make a meaningful contribution. And so our primary goal today is to inform you of the current organizational structure of CRIS, some of our planned, current, and future studies and ideas, so that you can understand how you can contribute to CRIS, and we certainly want to bring you on board. Okay? So, make sure this is working. CRIS, as you can see here, I tried to use the old Roman structure to capture what CRIS looks like. But basically, CRIS is a research cluster that consists of 3 distinct research pillars. And I'm just going to call them biomedical and agriculture pillar, which is the genome diversity pillar, and a policy and socioeconomic pillar. And these pillars are underpinned by a foundational pillar that we call the knowledge mobilization. Because we think in CRIS that the key role that we play is to mobilize our knowledge, because right now, as you're aware, there are a lot of knowledge gaps out there, both within the academic environment, in the industry environment, in the public environment, and in the government. So all our activities are going to be associated with mobilizing the knowledge that we gain as fast as we can so that our stakeholders can take advantage of that knowledge. And of course, all of this activity falls under a common roof that consists of our collaborative partnerships between industry, academia, and government. So that is the overview structure of CRIS. That has expanded since December. We were just primarily this, and after December, we became this. And we hope to see it continue to grow. So we are a research cluster, an interdisciplinary research cluster with 3 distinct pillars: biomedical, genome diversity pillar, and our policy and socioeconomics frameworks pillar. I'm not going to talk to you about those pillars because the individual leads are going to come up here and discuss what those pillars are. And I'm hoping that people in the audience today can see, oh yes, I can fit within that pillar, or I can span all pillars, or I want to contribute to the knowledge mobilization pillar. So we want your interest, and we want to capture you into CRIS. We have a website. It's admittedly, it's based upon the University of Saskatchewan website structure. We will be rebranding this website. We wanted to wait on that until we had further information about the contributions that others are going to make to this research initiative. And we really must be something because we even have our own Wikipedia page, okay? So you know you've arrived when you're in Wikipedia. So with that very short overview, I'm going to then turn the floor over to our pillar leads. Not all the leads are going to come up here and talk to you about their respective activities within each pillar. But first and foremost, we'll talk about the biomedical pillar. And then so I'll invite both Drs. Lepairie and Dr. Huntsman as spokespeople for the various activities that are going on in the biomedical pillar. And
Okay. Hi, everyone. So my name is Robert Lepairie. I'm an assistant professor in the College of Pharmacy and Nutrition. And I'm going to talk a little bit about the current activities of the biomedical pillar. And then I'm going to turn the floor over to Dr. Richard Huntsman, and he'll give an update on the clinical side of things.
Speak loud enough.
Oh, sure.
Make sure you get to the face.
This thing. I speak very quietly normally.
I know.
All right, so our goal within the biomedical pillar is to try and obtain scientific evidence about the application and the evidence-based medicine in and around cannabinoids. With that in mind, the pillar leads, myself and Dr. Alcorn, Dr. Mousseau, and Dr. Huntsman. What we're hoping is that we can look at these three key areas of biomedical research. So the first being just biomedical research itself, pharmacology, neurosciences, and biochemistry, and then clinical research, and Dr. Huntsman will talk a bit about the studies that we have going on, as well as I'll mention a few of the studies that we are trying to plan and get underway. And then finally, health services. So this is looking at assay development and therapeutic drug monitoring. We have a lot of active research going on in this pillar, which is really exciting. If you want more details, you're welcome to ask questions about this later. I can't get into all of it right now because it just might get overwhelming. But on the biomedical side, these are 5 of our main projects that are on the go right now. So we are developing novel ligands for the type 1 cannabinoid receptor. And we're also looking at lesser known phytocannabinoids. So these are cannabinoids other than THC and cannabidiol that are found in the plant, and trying to understand what the pharmacology of these compounds is. We're looking at treatment of absence seizures with CB1 targeted or type 1 cannabinoid receptor targeted compounds. We're working to survey the endocannabinoid system in Alzheimer's disease, and then we're looking at epilepsy and cannabinoids in the context of the gut microbiome. For clinical research, we have the CARE-E study well underway, and Dr. Huntsman will talk to you more about that. We also have studies looking at the impact of lipid and lipoprotein alterations, and then a new study just underway looking at pharmacokinetic evaluations of cannabinoids in pediatric populations. And then for health services, we're looking at the prevalence of prenatal exposure to cannabis. We're looking at several mobilization and knowledge translation activities, and we're conducting 2 systematic reviews of the literature to understand the knowledge gaps that exist for multiple sclerosis and dementia. So with that, I'm going to turn it over to Dr. Huntsman after I just give you a brief update and a shout out and let you know that Dr. Matthew Links is working on a study in a chicken model of epilepsy that we know to be responsive to THC. And we're hoping to use this as a new animal model here in Saskatchewan to better understand the usefulness of cannabinoids in the context of epilepsy.
So thank you. So my name is Richard Huntsman. I'm a pediatric neurologist in the Department of Pediatrics here at the University of Saskatchewan. And so what I've been asked to do is to give a little update on the cannabidiol in children with refractory epileptic encephalopathy study, which is a multi-center study being led through the University of Saskatchewan in Canada. I realize that there's a lot more people in the audience today who weren't here when I gave my initial presentation on CARE-E. So what I'll do is I'll just give a little brief, very one minute rundown of what CARE-E is. So what CARI is, it's an open-label dosage escalation study using Canamed 120, so 1 THC to 20 cannabidiol cannabis herbal extract in escalating doses in children who have medically refractory epileptic encephalopathy. These are the epilepsies which are just devastating in childhood. These kids will have 40 to 100 seizures a day. It's associated with developmental regression, and they truly are refractory to treatment. Most of these kids end up on multiple anticonvulsants, surgery, special diets to try and control their seizures. So we developed this study because there was 4 or 5 years ago in clinic with my patients with these epileptic encephalopathies, I kept on getting asked, what about cannabis? What about cannabis by the parents? And I'd have to say, I don't know. We just don't know. So what CARI hopes to answer as an open-label dosage escalation study is that there is a good growing body of evidence that cannabidiol and cannabinoids can be very beneficial in some of the children with these types of epilepsy. But there's a lot of questions we just don't know. Which kids respond? Why do they respond? What are the interactions with a cocktail of anticonvulsants that these kids are on? What about pharmacokinetics? What about, do we go about what's the most appropriate dose to use? Should we use just pure cannabidiol? Do we add a little bit of THC? These are all questions nobody knows. So these are some of the questions we hope to be able to answer with CARI. So with CARI, there are 5 sites that we are enrolling kids in. So in our Saskatoon site, we have actually completed enrollment and we have completed the study in the Saskatoon site. So these are 6 kids with epileptic encephalopathy. Keep in mind, most of these kids are on multiple anticonvulsants. The likelihood of adding another anticonvulsant and the child becoming seizure-free is about 2%. So, based on our data, as we escalated the dose of cannabidiol and we gradually built up to 10 to 12 milligrams per kilogram per day of cannabidiol, 2 of our participants became seizure-free, which is quite remarkable. One of these children is a patient I showed in the initial Café Scientifique, who had evolved into an EEG picture called continuous spike wave in sleep, which is devastating. Basically, when they fall asleep, they just go into a continuous seizure and almost impossible to treat. His EEG normalized, which was quite remarkable. Another 3 participants had a greater than 50% reduction of seizures, ranging anywhere from about 55 to about a 70% reduction in seizures. Interestingly, it seems to be for 2 of the children, motor seizures seem to respond the best, as opposed to what we call dialeptic seizures, where the child will just stop and stare and then come back. We saw a better improvement in kids who had motor seizures, which tend to be a lot more devastating. And then 1 participant had a 45% reduction in seizures. We had a mixture of children with epileptic encephalopathy, including children who had Lennox-Gastaut syndrome and an entity called Dravet syndrome, which is a very severe epilepsy. One of the nice things is that none of the kids withdrew due to side effects from the medication. And that's always been a concern, that, what about the THC? They're going to become stoned. They're going to become psychotic. We really didn't encounter that in our patients at all. The only issue we encountered is that some of the kids who are on a medication called clobazam, it's like a long-acting form of Valium we use to control seizures, we had to drop the dosage of clobazam because some of the kids got a bit sedated with the combination of the cannabidiol and the clobazam medication. But once we dropped the dosage of clobazam, that resolved. So where are we now? So, this study has been about, we talked about this 5 years ago. It's been a learning experience, but we are moving ahead, finally. So, as I said, U of S, we've got enrollment, all 6 kids have completed the study. And every child who completed the study, the parents requested that they stay on cannabis oil at the end of the study. So UBC, we got ethics and operational approval. It took us over a year to get this study through UBC ethics. Once again, you're giving them cannabis, you're going to make them stoned, they're going to get psychotic. But, so we had to fight with them to try and get this through. Finally, we've got approved. And we've got operational approval from BC Children's as well. We did a site initiation visit 3 weeks ago, and so hopefully we're going to be starting to recruit in the next week or two at UBC. They already have kids lined up, ready to go. University of Montreal, once again, ethics approval expected at the end of next week. Operational approval has been obtained at Hôpital Saint-Justine, and we have done the first part of a site initiation visit. The pharmacists need to have another sit down just to talk about dispensing the cannabis product. And the University of Manitoba and Alberta are in a similar situation. Ethics approval is obtained, and we're just waiting the final word from the respective hospitals to get operational approval. One of the big hang-ups has been getting the research pharmacies to actually hold and dispense the medication, because a lot of these hospitals, this actually goes against the hospital policy of dispensing cannabis products. So if you're in RUH and you're on cannabis, the nurses and the pharmacists are not allowed to give it to you. You or one of your family members has to administer it. So we were kind of going in, so it took a little while to work around that. And, and I jumped over back to Robert. So I guess, would you like me to talk a little bit about so there are some other studies which, from a clinical studies that we are looking at. So one of the questions which has been raised repeatedly is, what about pharmacokinetics? And as a pediatric neurologist, I can say that pediatric neurologists tend to be a very conservative group when it comes to medications and new treatments. And this is one of the major stumbling blocks for cannabis being accepted in the mainstream medical therapy amongst neurologists and pediatric neurologists. So we are doing a study looking at pharmacokinetics in children, different age stratified levels, for single dose study for kids. And also we're looking at, as well too, I'll go back on to Robert's side too, but we're also looking at some other studies as well too, looking at a prenatal exposure, because that's a big area of concern for us as well too. And hopefully we might be able to shape some policy with regards to recommendations about cannabis use in pregnancy as well. And I think I'll hand it over. So,
Thank you. All right, so I'm just going to pick up exactly where Richard left off and talking about some of the stuff looking forward. So if you were at our last Café Scientifique in December, you might have noticed that a number of the things that we talked about that were in the planning stages in December are now well underway. And now we have even more things to look forward to. So on the biomedical research side, we have studies that we're looking at initiating for infantile spasm, as well as multiple sclerosis, and then developing some radio-labeled tools that would allow us to track some of these cannabinoid compounds in living systems. For clinical research, we're hoping to expand out the CARE-E trial to more sites, as well as looking at different dose escalation opportunities in dementia, as well as in cerebrovascular physiology studies for concussion and traumatic brain injury. And then we're also interested in investigating cannabinoids in companion animals as a veterinary medicine application. And then finally, on the health services side, we're looking at developing knowledge user surveys to help understand where people are at in their own education and use, as well as developing tools and mobile apps that would allow people to ask questions about their use and how it's affecting their well-being. Hello. All right. During the last Café Scientifique, we talked a little bit about an NCE, or a National Centers for Excellence opportunity called Cannabinet. That was going to be a national network that it would link up all of the different cannabinoid researchers across the country. Unfortunately, our LOI was not invited forward to submit a full application, despite the fact that we had over $25 million of funds raised across the country. However, money's not just going to disappear, so the people who got together to form Cannabinet are going to be meeting at the beginning of April in Toronto, and we'll talk about next steps moving forward and hopefully get something moving on this. So then I wanted to talk to you a little bit about partnerships, because we've been really successful as a group in orchestrating partnerships, in acquiring funding for studies, and I think this speaks to how much we have going and how much we can continue to have going for CRIS. So these are some of our current funding partners: the Children's Hospital Foundation, SHERF, Innovation Saskatchewan, NSERC CIHR. Canamed has been very helpful in getting some of our studies underway and collaborating with us in an open and scientific environment that's been really positive. Several colleges from the University of Saskatchewan, as well as the University of Regina, and new collaborations that we're establishing with different partners at the University of Calgary, Dalhousie University, and now the University of Melbourne, and maybe some others in Australia coming soon. We also have preliminary or early days talks with two other companies, Sundial Growers and Greenhouse Ventures. Those are early days, and that's looking at some of the educational pieces around cannabinoids. And then we've established a partnership with the CCIC, or the Canadian Consortium for the Investigators of Cannabinoids. And this is a big national group that's looking to provide resources for highly qualified personnel, trainees, graduate students, as young researchers and scientists. We have a meeting coming up to try and bring all of this together and provide knowledge mobilization, specifically in this case to healthcare practitioners. So this will be our inaugural Saskatchewan Cannabinoid Research Symposium. We'll be hosting a 2-day symposium on August 16th and 17th at Marquis Hall right here at the U of S. Registration will be free, and our goal is to provide filtering of anecdotes for evidence-based medicine and showcase the HQP and the research that's already underway through CRIS and through our collaborative efforts. So that's all I have, and so with that in mind, I'll turn it over to Tim, who's going to bring up the next piece. Thanks. I'll load your slides for you.
Oh, sure. Okay, thanks. So I'd like to thank the organizers for inviting me to give a talk. I don't do cannabis research, although I'm entering the field. So what I wanted to show you today is how we will contribute to cannabis research, and we do two things. One is I'll talk to you a little bit about breeding and reproduction in plants. And then I'm going to show you how we can find genes. And so I run a research group here. I run a research group in Germany. We've been funded by European funders. We're working on dementia. We're working on things like chamomile. So I'd just like to take you through some of our studies. I'll take about 10 minutes of your time just to show you what we can do and how we would like to apply this to cannabis research. There we go. All right. So we study reproduction. I'm a molecular evolutionary biologist. So, and I train a very large group of people. And what we study is reproduction in plants. So I'm assuming you guys can hear me. I spent the last 10 years over here at one of Europe's largest plant genetics institutes. So it's a Leibniz Institute for Crop Plant Genetics and so plant genetics and crop plant research. It's also Germany's largest gene bank, and it's one of Europe's largest gene banks. So gene banks, of course, contain all of the seed material for our crops and relatives. And right now, for example, there's big pushes across the world in different gene banks to sequence all of the material they have there because, as we all know, we tend to live in a monoculture world when it comes to crops. And so what we're interested in doing is harnessing biodiversity and close relatives of crops to try to bring in genes for resistance to things like abiotic and biotic stress. And so 2 years ago, I came to the U of S. I brought my team. So before I go anywhere, of course, I have to thank these guys because these are the guys who do the work and generate the data that I'm going to show you. So as you can see, this is my group in Germany. And as I got to the U of S, we've expanded quite rapidly. So I've got a lot of expertise in plant reproduction and so on. Here we go. All right. Quick historical trick. So, how many people know Mendel? All right, you guys and we all learned about peas with Mendel, right? So Mendel did the crosses and so on and saw segregation, and the first law of genetic segregation was developed, right? Very happy, published this paper and so on. Then he thought, well, I'm going to try it again in another plant species, and he chose Hyraceum. So it's, why can't I remember the, okay, I can't remember the natural common name doesn't come to me for some reason. Okay, suffice it to say that he crossed Hyraceum in the pea gardens in Brno, and sure enough, when you looked at segregation in the F1 generation, there was no segregation. And he couldn't figure this out. And he couldn't figure it out until he came up with a second law of genetic segregation. And so basically, I won't even go into it, but basically trying to describe this observation that that segregation didn't occur as it did in peas. And what we now know is that what was happening in Hyraceum was a phenomenon called apomixis. So apomixis is a form of asexual seed production which occurs naturally in plants. And before I just explain that, so of course, you know, your point of reference is sexual reproduction, and I think every one of us here understands sexual reproduction, where you have a mother and a father. Both of those things produce meiotically reduced gametes. Those gametes come together to produce offspring, which are 50% related to the mother and father. In apomictic plants, now remember, this is a natural form of reproduction. What's very interesting is that female plants will produce an egg cell, which is meiotically unreduced. So it's a genetic clone of themselves. And that egg cell will develop parthenogenetically, so without any fertilization, no contribution by the males. Males are useless in this population, okay? We won't go into that. But the point being is that these plants will produce, the mother plant will produce genetic clones of herself. And furthermore, what's very interesting is these clones can be very complex genotypes, right? So these are not like inbred lines or anything like that. These are complex genotypes which are somehow meiotically frozen and then passed through time. So why is that important? Whoops. Well, actually, sorry. Go back one. There we go. So it's actually kind of interesting because there's a large number of animal and plant species that do this. And very interestingly, in all of these cases, their ancestors were sexual. And so what we do in my group is we use evolutionary theory to try to understand why this switch from sexual reproduction to apomictic or asexual reproduction was made. And we use this theory to actually identify genes underlying this reproduction. Now, why is that important? Well, it's important if you think about crop production these days. A lot of our crops, now this will also apply to cannabis, okay? So a lot of our crops are based upon the phenomenon of hybrid vigor. And so the idea being is that large companies, so your Monsantos and the Pioneers and so on, they spend a lot of time and effort inbreeding plant lines, and they'll inbreed them for 8 to 10 generations, which takes a lot of time and resources. These homozygous lines that have been inbred for repeatedly are then crossed, and they produce a first-generation hybrid. So this is the seed that goes to farmers. And those plants will be uniform. So if you look at a field of canola or maize or something, all of the plants are the same. And then secondly, they exhibit hybrid vigor. So they're bigger, better, higher yield than parental plants. Now that's the way it works. And if you're a farmer, right, and you bought the seeds from the company in the first year, your plants grow beautifully, and you want to get them out into the second year without buying seeds. So you cross your plants. And then what happens is sex and meiosis just mixes everything up again. And if you take your F2 generation, you lose all of that vigor, you lose the uniformity and so on. So what it means for the farmer is the farmer has to buy seeds every year. And the idea that we work on in my group is if you could imagine sex as a switch, you could just turn it on and off. What we could do in this F1 generation, which is a complex genotype, is that you could freeze it. And then you could proliferate those plants forever and ever. And so what it means, like if you were a farmer, is that you would buy seeds one time and you would no longer buy seeds in the future, right? So, I mean, it sounds bad, but there's actually a lot of benefits. And this is what drives us, right? So apomixis would completely disrupt breeding in plants forever, because right now the way it works is because the companies take so much time and effort to generate these inbred lines, you got to do the crosses, and every time you do one of these steps, you run into things like genetic incompatibilities and so on. So at the end, you kind of get a one-size-fits-all crop, right? And those are the crops that we see growing across the world. And what we're doing is we're missing all of this genetic diversity, which is available to us, but it's unexploitable because meiosis doesn't allow us to bring it into our crop species. So diversity is definitely what's driving us to do this, because there's a lot we can feed the world. We can create plants that are resistant to stresses. We can create plants that have good biochemical profiles and so on, but we don't have the genetic tools to the ability to proliferate these on an industrial level. And if you look at publications and so on, people tend to put the term billions in terms of the change it would have in various plant industries. Okay, now why am I telling you all that? So that's the bread and butter of my group, okay? And a couple of years ago, we found a paper from the 1930s in a German journal, and a guy, someone described apomixis, so this naturally occurring phenomenon in hops. Now, hops, hemp, and cannabis are sister species, really, really closely related to one another. And so we've got these things growing here at the U of S. And so one of the things we would like to do in this consortium is to breed or somehow harness apomictic reproduction, this asexual reproduction, to take it from hops and to transfer it into hemp and into cannabis. The idea being that if we could do that and we could have that switch working in them, then we could begin crossing. We could cross cannabis and hemp. You could cross anything you want, right? The point being is that we could begin producing a lot more biochemical variability that would go into into the medical studies and so on, to provide more fodder or variability to generate kind of the signals, like, what are the active compounds? What are the combinations of compounds that give us the desired effects in human beings? Okay, and then so the final part is just, now, so I talked to you about reproduction, right? But what about medicinal plants? Well, what we do in my group is we study naturally occurring apomictic species. And so these are natural populations of plants that are composed of individuals that reproduce sexually or reproduce apomictically. And interestingly enough, so these are some of the species we work on in my group, but interestingly enough, you'll see some things that are actually medicinal. So I'll just take you through two quick things. So I'm part of a €2 million consortium in Germany, right? Well, actually, it's European now. And what we're doing is we've found that in Hypericum, so St. John's wort, that there are actually compounds in St. John's wort that can fight certain symptoms of dementia. Now, one of the symptoms, before I get to the dementia symptoms, let's just take St. John's wort, okay? We'll start, this is just kind of like a case study, and this could be like cannabis. So, we've collected St. John's wort from all over the world, all right? We've got about 2,000 accessions of things. So this is what we do, is we collect St. John's wort, in this case, this could be, and this will be cannabis in the future, and hemp and so on. But what we can do is characterize the genetic backgrounds of these things, right? So plants are in an evolving world. There's a lot of genetic variability, and sort of superimposed upon that genetic variability is biochemical variability, right? So that means if you are doing a medical study on something, on a particular compound, well, it matters if you pick a plant that's from here with this blue genetic background versus a plant that's over here with a red genetic background, right? So each of those plants produces a different kind of biochemical profile, which is influenced by both the genotype of that individual and the environment in which that individual grows. Okay, so that's the resource, and that's what we can do in the cannabis and hops and hemp research field. So with Hypericum, for example, with St. John's wort, we published a paper a couple of years ago where we can actually show in mouse models that very crude extracts from St. John's wort lead to an increase of transport across the blood-brain barrier, which is, in mouse models, it's one of the models for one of the symptoms of dementia research. And so hence, in this particular consortium now, we got €2 million. And what my group is doing, so this just shows activity, I'll leave that alone. So what we do in my group is we go back to these different genetic backgrounds of St. John's wort. Once again, we'll do this with cannabis. We can sample flowers at different stages. We can do genomics on these flowers. We can do transcriptomics and so on. We can really get into the reproduction of them. But in this particular case, what we were able to do is in a case study where we did this comparative genomics and biochemistry of these different genetic backgrounds, we were able to identify genes for biosynthesis, so things that are involved. So I won't tell you what those compounds are, but the compounds that lead to the increased transport across the blood-brain barrier, we've got candida genes. We've identified very strong candida genes associated with the biosynthesis of these compounds, in addition to the organs in which those compounds are produced. So we can find those things, right? And those are the kinds of things we want to introduce into cannabis, into cannabinoid studies. The second one, and I'll just do this really quickly, is chamomile. So I've been funded for almost 10 years now by German chamomile producers. So medicinal plants in Germany are huge. It's a really big industry compared to what you find in Canada. And so chamomile, for example, is a really well-known plant in Germany. It's harvested. I mean, it's not an enormous crop relative to things like canola and so on, but it's still, it's a major crop. So we're being funded for the same kind of thing. So, so we go into chamomile populations. We've collected chamomile from all over the world. We can do the biodiversity of chamomile. All of these. So if you can read a phylogenetic tree, what this group of things, these are all closely related accessions of chamomile. These are the things in production in fields, right? And so these are the things that are being sold to consumers. And then all of this is unharnessed genetic diversity, which can also go to the consumers, right? So we're interested in characterizing that and quantifying that for the industry. And so one of the things we're interested in is male sterility. So crossing plants is of utmost importance no matter what kind of agriculture you're doing, right? So with things like with chamomile, we're interested in understanding genes that could, for example, turn off maleness or turn on femaleness and so on. So what we can do is comparative genomics and comparative transcriptomics on plants that either have both male and female flowers or female flowers only, and we've also got some species that only have female flowers. So you can do comparative studies to identify genes that you can actually use for reproduction. Okay, and then the final thing is that, this is a study we published last year, and so alpha-bis-albolol, I'm almost done, is a compound that is associated with digestive function in human beings. So it's a compound that helps with digestive function. And what you can see, for example, that if you look at different genotypes of chamomile, for example, you can have different levels of this particular active compound. We expect to find very similar things in cannabis and hemp and so on. So basically, when we think about molecular markers and breeding, right? This is where you are, where the medical researchers are going to be, right? So you want to, and you can correct me if this is just my, view of the world right now. But essentially, we're looking for the best plants that produce, particular targeted biochemical traits or profiles which are associated with a particular human trait, and that could be leukemia, that could be, leukemia, it was dementia or epilepsy and so on, right? So these different biochemical profiles will have different kinds of effects on the patients, right? So and this is basically how we see it. So we'll start off with all of that diversity, right? And so I've already been working on getting germplasm from various colleagues all over the world. We're going to have one of the largest cannabis germplasm collections in the world here at the U of S. Once we have that, then as a group, what we can do is assess the biochemical and genetic diversity of that resource, and then from that, we'll make choices of things that will go into these other studies with our other colleagues in this pillar. So with that, I think I'll just stop. Yeah, that's it. So this is where my institute was in Germany. And I'll thank you, and I'll leave the podium to Jerome.
Thanks, Tim. That's a great background for some of the discussion that we're going to be having related to some of the policy challenges. So from a policy perspective, I believe that a successful policy model or approach starts by clearly understanding your policy objectives. And I think Peter has already alluded to that. But what often gets missed is evaluation to see if the choices you made are actually giving you the desired results that you had wanted to when you started out your policy. And so this, in a nutshell, really describes our approach to policy and the way we think it should be and what the policy pillar really wants to tackle. Now, what's unique and interesting about this whole policy question and framework is let's revisit some of those policy objectives. So Peter had outlined this. One of the policy objectives is to reduce consumption of marijuana in the youth population. The second one is to reduce the illicit market. And the third one is public safety and public health. Now, a fourth one that's often alluded to is the economic benefits of this. So what you have in those three creates a really interesting complexity for starting to establish policy frameworks, because you can start to see that the choices you make regarding certain policy decisions will impact more than one of those. So for example, let's just choose something that's been topical lately. What about the age? So if you choose 21, what impact will that have on illicit market? Will people continue to use the people? And just so you know, some of the studies indicate that the second largest user group of cannabis in Canada is between 12 and 17 years old. So are those people just going to quit using marijuana? Now, the issues from a medical perspective is, and the arguments that were made by the Saskatchewan Medical Association, is that it affects brain development. And brain development is widely accepted, that 25 years of age is widely accepted as the time period for brain development up until that time. So you make the choice. Do you make a choice to say, okay, we're going to minimize the effect on health because we're going to go with 25 or 21, but what impact is that going to have on the illicit market and youth consumption? We don't know the answers to those questions. We can look at the choices that were made in other jurisdictions. We have Colorado, we have Washington, we have Oregon, and other jurisdictions. I believe Uruguay is the only country that is completely legalized. So you can certainly look at that, but then you're dealing with a different culture, a different population. And also in Colorado, the numbers will be skewed by the fact that Colorado is surrounded by a bunch of states and regions that do not have legal marijuana. So what impact is that having on the users and the consumption and the sale in Colorado? Does that skew the numbers? So where I'm going with all of this is that I think one of the most effective and most important things to look at in our policy framework is metrics. Yep. Okay. I'm looking on the wrong side. Okay. So I just reviewed again the policy objectives, and let's now go on to the multidimensional. So we talked about this multidimensional nature. I know one thing I forgot to mention in the complexity is that you now have 3 levels of government involved in the policy development. You have the federal government, which is passing the initial legislation expected sometime this summer. The date is still open-ended, but they expect it sometime this summer. The provincial government, and then municipal governments. And those choices, if you think it's difficult to get the federal government, the provincial government, and then you add municipal governments to agree and to coordinate their policy choices, well, you're a better man or person than I am, because it's incredibly challenging in the whole innovation space, trying to coordinate the activities and align the different choices that are made. So we have a real complex situation. Added to that, at the provincial level and at the federal level, you now have a number of different ministries that are involved in this, and they all bring a different perspective. So you have health, you have corrections and public safety, you have agriculture, you have the economy, you have post-secondary education, you have Innovation Saskatchewan, and there's probably some other ministries that may be interested and engaged in this. So you now need to get teams together that are starting to understand what impact is this going to have on enforcement, for example. If we make a choice, what are we missing? What do we need for enforcement? And right now, the biggest challenge in enforcement of not only the regulations, but impairment, is the lack of a reliable impairment tool that will give you definitive data that will work in the courts. In some of the off-the-record discussions I've had with RCMP, they said, you know what, we can't count on the results. If you do, they're looking at a swab for data, and it'll indicate whether there's THC present in the sample. But what you don't know, because THC is fat soluble, is how long it's been there. It could have been there a couple hours. It could have been, 20 days, 30 days. I think it's up to a month, I believe, that it will accumulate. So, and what is the standard for impairment? We all know the issues. We've had, how many years of alcohol been illegal, and we still haven't figured out what the impairment level of that is. It's gone from 0.08 to 0.4 and 0.04. And so we have real issues related to that. Not to mention that impairment is genetically, there's genetic variabilities. We all don't react to these bioactives in the same way. So what do? You take the lowest common denominator. Our government has already said that any presence of cannabis in the system is going to be considered impaired. So what does that mean? What are the implications of that? So what we have is an extremely complicated regulatory framework, and how do we manage that? So we look at some of the areas that we've talked about. Now this is an open-ended, so this isn't definitive. We're not saying this is it, this is where we're going to research. We're throwing this out based on the discussions we've had internally in Johnson Shoyama with Kathy McNutt, Peter Phillips, Ann Valentine, and myself, and other discussions. We've identified some of these areas as things we'd like to tackle on the policy framework. The sustainability of the supply chain. And after you heard Tim's discussion, you could see that what you're going to need is an identity preserved or a closed loop system. While having genetic markers that can help you identify very specifically which variety you're dealing with is really important. And one of the reasons it's important is there's a hemp industry that is starting to grow in Saskatchewan and creating a lot of economic benefit for farmers, and the applications for the products from hemp are quite wide-ranging. And what impact will this have? Right now, the definition of hemp is 0.04% THC or less. And there are hemp producers who have their own breeding programs, and they're way below that. And they're working very hard, spend a lot of money to keep their levels down as low as they possibly can. In fact, they say they have some varieties that have virtually almost zero THC in it. So they're very concerned about choices that would be made in terms of how is the government going to regulate varieties? Is it going to be registered seed? If people grow it at home, the risk of contamination with the hemp crops. If people start growing marijuana, cannabis outside, it's going to have a real impact on the hemp growers. And you have the issues that you have in other crops. For example, mustard. The mustard growers were very concerned about GM canola. So again, there had to be regulations, there had to be studies done on pull and drift to see how far is there a safe margin in which you can grow these crops so they can coexist peacefully. So Johnson Shoyama has engaged some of the hemp producers, and we're working on a seminar, symposium involving hemp production, and just having people from that industry talk about what are the possible implications for them and their business in this whole thing as marijuana becomes legalized. So this whole idea of identity preserved system. Now, I did work for a company that processed hemp seeds. About 10 years ago, I was involved in this, and we had to get approvals. And what made our approval process challenging was every board member and every corporate officer had to go through a security clearance. And 2 of our board members were Americans. So where do you get the clearance? Is it the American government or is it the Canadian government? Well, the Canadian government accept an American, and then you have to pay. By the time we got, all of our board members got the security clearance, we had to start all over again because the time was up, and we had to do it over again. So what we did was we basically created a spin-out company. 2 of the operators who worked in our processing plant, they were made the board and the corporate officers. So we had 2 people who were from Saskatoon that went down to the police station, got their security clearance, and that's how it was done to cut through some of the the issues that we were having with the practicality of implementing a policy. So this whole identity preserved and globally system, all of these areas include great opportunities for innovation. You know, so just the whole genetics and the development of different varieties with different bioactive profiles are really quite interesting. And if we start looking at some of our clinical research, can we purify so a plant only has THC or we can isolate THC or CBD and do studies on that, and then do combinations, formulations to see is the therapeutic benefit derived from one active or is it a combination of the actives? And there's other cannabinoids that are involved. So what would that do? So doing clinical studies where you actually have different groups that are tested, and then you can look at some of the comparisons. Might be an interesting area in terms of you can then start to get intellectual property for a variety. If it's proven clinically that it has this benefit, then you can get the IP to protect it for that market application for that benefit. And I think if Saskatchewan is looking at a vision for innovation, this is an area where we should be considering. We can be unique in the world, so we've got a variety. We have all the capabilities with our Crop Development Center and molecular biologists like Tim to actually develop a variety of new varieties that have very specific profiles and different features. So one of the things, they even looked at some phenotypic, and I think, Larry, you're the one who probably told me this, but phenotypic variability in terms of the size of the plant. So it's better if you're growing them indoors to have smaller plants, right? A little easier to manage. And those are some of the things that could be possible. So each one of these areas of research often will yield many opportunities for innovation. Safety and health, the quality control and regulatory systems are going to be interesting. So we need to have tests then to know what people are, when consumers buy it, what are they getting? In the company I worked for, we were an innovator in the industry. We were the first people to do voluntarily certificates of analysis. So we would do an independent lab, we'd use the Saskatchewan Research Council and a POS pilot plant. We'd do tests that would say, this product that you're buying from us is guaranteed to have this bioactive profile. You're getting 18% DHA and 12% EPA, or the other way around. Those are the kind, and so the customer knows that there's some sort of verification and validation from a credible organization that says this is what I'm buying. You don't have that issue in the illicit market. You know, I don't think people buying marijuana on the streets are given a certificate analysis saying, by the way, here's how much THC is in the sample you're buying from me. So I think those are the kinds of things that are going to become critical. And if you're looking at the consumer, I think it was Deloitte's study that said there's up to 17% of the people who responded to their survey who said they might try marijuana. Do we understand what would tip them over the edge to be a user? Is it guaranteed quality? Is it safety? Verification that it is in fact safe? We need to get a better handle on that, so then again we can make the choices that are going to in fact grow that market. And public engagement and decision making is another critical component of this. So how do we get the data, evidence-based data, to make the choices that we need? And I think that's why I really believe the metrics. And Peter, I'm going to ask if you want to contribute some thoughts on them on the metric side, because you're much more
conversation.
Yeah, okay. He has much more background in metrics than I do. But I think this whole area of now, are we going to get politicians, once they establish a policy framework and data, evidence is developed that suggests that choice isn't working? In other words, you chose this age limit, it's not having the desired effect on reducing the illicit market. Are you willing to change? We hope they will. And so what we've always proposed is evidence-based policy development. And that's why the connection between the policy people and the clinical and the plant producers, the plant biologists, is all critical. They're all interrelated, which is why we were very supportive of our participation and involvement with Chris. I think it's absolutely critical that we're all involved and that we encourage our government colleagues and the politicians to adopt the same approach of collaboration and cooperation and sharing information. So it's one thing to generate a lot of tax revenue, but where is that money going to be spent? How much will they invest in research? How much will they invest in enforcement? And how much will they invest in public education and awareness? So those are things that, as policy developers, we need to be asking and holding our policymakers accountable for. And so we have actually established a report that was issued last fall. It's a good start. My piece was more or less on the innovation side. But I encourage you, if you haven't got a copy or haven't seen it, there are some outside to pick up a copy of that, as well as a coffee if you want. Pardon? They're online as well, yeah. So I'm going to turn it back to Peter. And Peter.
So as a final advertisement, we do have policy briefs. There are a couple out there. The rest are online, including the policy document that you heard about. And this lecture was recorded, and it will be judiciously edited to avoid some of my foibles, and put on the school website and I believe Chris's website as well, to assist for people who want to go back and use it as an aide-memoire or want to refer to someone else for their education and training. Thank you.
Thank you. Thank you.